Today, I'll be speaking on the emerging role and future direction of CAR T cell therapies. I have no disclosures. And for the objectives, I'll review the current status of CAR T cell therapy in pediatric ALL, review the limitations and active efforts to address these challenges, and discuss future directions and new efforts in hematologic malignancies. So, everyone is familiar by now with CD19 CAR T cell targeting. What has made CD19 CAR T cell targeting such a success is that CD19 is ubiquitously found on B cells. Showing here a standard schematic, you have your CAR T cell with your T cell, the co-stimulatory domain, and the scFv, which is targeting your B cell antigen, in this case is CD19. You can see here that CD19 is nicely broadly expressed through the range of B cell maturation. Today, I'll be talking briefly about CD19, but also we'll focus a little bit on CD22 and talk about alternate antigen approaches. Now, our very first experience with CD19 CAR T cells in the pediatric oncology branch at the NCI was in July of 2012. In this case, we used the CD19 28-zeta with the FMC63 as the scFv for CD19 CAR, and this is now the Yescarta construct. Our trial was a phase 1 dose escalation study in children and young adults with pre-B ALL, and this initial experience was what allowed us to learn about the management of cytokine release syndrome, allowed us to understand the differences between CAR T cell persistence and knowing the impact that a 28-zeta co-stimulatory domain may have compared to a 4-1BB. We learned through this trial the importance of fludarabine and cyclophosphamide for conditioning chemotherapy. We were able to, in the context of this trial, treat patients with CNS disease and we're able to show that we could effectively and safely treat patients who had active CNS disease. And with this CAR being a less persistent CAR, we also learned about the role of stem cell transplant to consolidate a remission. And it really, in terms of our overall outcomes, you can see here we have several patients out who are now out, well beyond 5, 6, and 7 years. Our updated publication is now in Press and Journal Clinical Oncology. And overall, for us, this really helped to shift the paradigm for outcomes related to phase 1 trials regarding response. Prior to CAR T cell therapy, you were looking at a phase 1 trial primarily for safety, toxicity, and dose finding, but had very little expectation in the way of response. With these CAR T cell therapies, we were now starting to see response rates in the 60 to 70% or higher range, which really shifted what we think about as a phase 1 trial in patients with relapsed refractory disease. Now, the CD19 CAR T cell construct that most folks are familiar with is Kymriah or tisagenlecleucel. This is the 4-1BB CAR T cell construct that was developed at the Children's Hospital of Philadelphia that has now received FDA approval through the Novartis and the Eliana registration study. Shown here are the results. They found that there is an 81% complete remission rate in children with relapsed refractory B-cell ALL. And the event-free survival at 6 and 12 months was 73 and 50%. As I mentioned, this ultimately led to the FDA approval of tisagenlecleucel for pediatric B-cell ALL. Now, there are a host of CD19 CAR T-cell constructs that are FDA approved, including Kymriah for diffuse large B-cell lymphomas and 3 other CD19 CAR T-cells for adults with lymphoma, but there remains only one for pediatric patients. Now, the question really is, will CD19 CAR T-cells be the cure? This is the first patient who was treated with CD19 CAR T-cells and did not receive any additional therapy, who is now cancer-free for 8 years. And of course, everybody wants their child to be like this first patient, to receive CAR T cells and have that be the cure. The problem herein lies in the fact that this will not be the case for almost 50% of patients. You can see here that within the first 12 months, there's a pretty significant drop-off in patients who are going to relapse, which brings us to the current challenges. I like to divide these up into 4 categories. thinking about CAR T cell product failure and an entire science devoted to trying to think about how to get CAR T cells to be more functional or how to optimize the apheresis so that you can collect CAR T cells, especially in patients who've received heavy prior chemotherapy. This will continue to be an ongoing challenge as we try to treat patients who have received even more intensive therapy, as with AML. There's a concern for antigen loss, which I'll talk a bit more briefly about in more detail. In this case, you have CAR T cells that are active and effective. However, the leukemia cells are losing the antigen and thus they're evading effective therapy. There is a concern about CAR T cell toxicity, either in terms of severe cytokine release syndrome or neurotoxicity, now termed as ICANS. And the point is that you have to be able to survive the toxicity in order to achieve the benefit. And then, of course, a whole category of unmet needs. While CAR T cells have been highly effective in targeting B cell hematologic malignancies, there remains a lot of opportunity to improve this strategy in solid tumors and in other malignancies. To talk briefly about CD19 loss. So CD19 loss or downregulation represents the primary form of treatment failure. Walking you through this graphic, here is a leukemia cell with lots of CD19 antigen on the cell surface. In this case, this is your CAR T cell. You have antigen recognition, T cell activation, and tumor cell killing. In the case of antigen escape, you no longer have CD19 present on the cell surface, which leads to no T cell activation or tumor cell killing. There's also lineage switch, in which case leukemia comes back as a different lineage altogether. For instance, ALL comes back as AML, where again, you have the same problem where you cannot have effective T cell targeting. And then, of course, there's also antigen downregulation, where you may not have as many markers on the cell surface, but then this leads to an incomplete and an inadequate T cell response and insufficient tumor killing. So recognizing that single antigen targeting strategies may have limitations, one thought was that you can target an alternate antigen may help circumvent CD19 loss. So a large part of my focus in the National Cancer Institute in the Pediatric Oncology branch has been to move forward efforts on CD22 CAR T cells. This CD22 CAR T cell construct was developed in the Pediatric Oncology branch in the Mako lab, and we initiated this trial in 2014 as a phase 1 dose escalation study. We have now enrolled over 80 patients to date, have demonstrated that CD22 CAR T cells are highly active in patients with relapsed refractory disease. We found overall that there is a 70% complete remission rate, and that at the expansion dose level, we had a 76% complete remission rate, with the vast majority of those patients achieving MRD negativity or minimal residual disease negativity as detected by flow cytometry. We found that it was effective also in those who did not respond to prior CD19 targeted strategies or those who had relapsed after CD19 targeting. We found that the toxicity from cytokine release syndrome was overall quite mild, as was the neurotoxicity. While I won't go into the more extended toxicity profile for today's talk, what I will say is that the CD22 CAR T cells, we did see a heightened inflammatory set of toxicities that were associated with the macrophage activation syndrome or hemophagocytic lymphohistiocytosis-like syndrome or HLH-like toxicities, which is an area of active interest that we're focusing on that allowed us to see that different CAR T cells are going to have a different toxicity profiles. And then shown here is our waterfall plot for overall responses. With CD22 CAR T cells, however, we have the same problem where antigen modulation was a frequent cause of relapse. CD22 site density was lower than what we see with CD19, making these leukemia cells even more predisposed to relapsing after effective targeting. So in this case, we use CD22 CAR T cells for remission induction as a bridge to transplant to prevent antigen modulation as a cause of relapse. With allotransplant being an acceptable practice for patients for curative intent, especially for those with relapsed refractory disease, and knowing that we're seeing more patients who had not received a prior transplant, we're currently using it in that setting. Shown here is our overall swimmers plot of patients who are in a duration of a continuous remission. We have one patient who's not received any additional therapy, but most who are long-term survivors have received transplant. And again, we see relapses happening pretty early on, within about 4 to 6 months due to CD22 antigen downregulation. Based on our efforts, however, we did receive FDA breakthrough therapy designation for the treatment of pediatric and young adult patients between the ages of 3 to 30 with CD22 positive B cell ALL that is refractory or second or later relapse, that is either CD19 negative or relapse refractory to CD19 targeting. But knowing now that antigen escape is a problem with single antigen targeting strategies, we then were able to take our effective CD19 and CD22 CAR T cell approaches to then think about combinatorial or simultaneous antigen strategies as a mechanism to prevent antigen escape. Indeed, the foundation of ALL therapy is based in combinatorial treatment strategies. So why would we think that immunotherapy would be any different? Shown here are 4 different ways of being able to do combinatorial treatment strategies. You can do co-infusion, which of course would be quite costly as you have to have 2 different CAR T cell products. You could do co-transduction, which again, you still have to account for cost, but may have some limitation, the cell surface expression. We are currently testing this bivalent model, which is this loop CAR, which shows the scFvs in tandem, and that is currently out in the clinic. And pretty soon later this year, we'll have a bicistronic CAR, which is one vector that goes into a T cell, but then has an internal cleavable linker leading to equal expression of both CARs on the cell surface. As I mentioned, we are actively enrolling on a phase 1 study of a novel CD19, CD22 CAR T cell construct with the idea that it may diminish the risk of antigen escape. Shown here are my colleagues, Dr. Shalabi, who's reading the clinical trial, and Haiying Qin, who designed this construct. Shown here is the preclinical activity of bispecific CAR T cells. You can see here is a CD1922 CAR T cell construct. And preliminary data suggests safety and efficacy, although there are some limitations to its efficacy with CD22 targeting, and thus the next construct is already being planned for translation later this year with improved CD22 targeting. A new CAR T cell trial that we're actively working on that we're also hoping to open later this year is a TSLPR CAR specifically for patients with CRLF2 rearrangements. Patients who have CRLF2 rearrangement represent an extremely high-risk population of pre-B-ALL and represents about 50% of patients who are deemed as Philly-like. This is associated with JAK mutations. These are patients who are either have Hispanic or Latino ethnicity, patients with Down syndrome. In this case, the outcomes can be quite poor. Patients with a CRLF2 mutation have 35 to 71% relapse-free survival, 35% representing the CRLF2 population. And TSLPR is a cell surface marker in patients with CRLF2 positive BALL. You can see here our outcomes. This is a TSLPR in the cell surface. You can see here the site density looking at 3 different cell lines. This is the REH transduced with TSLPR CAR. And this is comparing outcomes of CD19 CAR, 2 different doses, CD22, and a highly effective TSLPR CAR. And again, we hope to bring this trial forward later this year as well, and we'll ultimately plan to put it into combinatorial treatment strategies. Now, going beyond B-cell ALL. So thinking about AML, this is an area where really more efforts are needed. Approximately 60% of children and young adults with AML will not achieve long-term durable remission. Considerations for CAR T-cell therapy in AML, however, are intrinsically more complicated. There's considerations for for antigen expression and whether or not there's more heterogeneity, making there be a greater concern for antigen loss as a reason, or downregulation as a reason for incomplete response to CAR T-cell therapy. We worry tremendously about the on-target and off-tumor toxicity of targeting hematopoietic stem cells, and then also think about a bridge to transplant. We are currently working on a phase 1 study of CD33 CAR T cells for children and young adults with relapsed refractory AML. And shown here is a treatment schema. This is a multi-center trial that incorporates the lintuzumab as CFD with CD28 costimulatory domain, and we have enrolled and treated 3 patients to date. Our next construct will be looking at targeting FLT3, which is the most commonly found mutation in AML with cell surface expression seen. And again, there's preclinical data for FLT3 CAR targeting is promising. And we're hoping to take this out to the clinic at the end of this year. And then finally, to end on challenges, is the question of what do we know about subacute or other long-term effects? I have put together a CAR T-cell therapy beyond the storm consortium to think about the toxicities, especially if these children and young adults are receiving these therapies for long-term cure. This is a multi-center, multidisciplinary group of care providers who are all well-versed in early implementation of CAR T-cell therapy. And we're working on a series of retrospective and prospective protocols to study subacute and late effects of CAR T-cell toxicities in children and young adults. And so with that, I think the next generation ideas is really to address the current challenges in CAR T-cells. We're thinking about persistence and regulation of CAR T-cells, antigen escape, decreasing toxicity, optimizing outcomes in other diseases, better targeting. I did not talk about solid tumors, but again, there's a whole area that really needs to focus on optimizing that therapy, off-the-shelf approaches, and of course, overcoming disparities with access to care. In conclusion, there are many, many people to thank, but I'm really grateful to be working with my collaborators here in India with Dr. Narula and Dr. Purwar. Thank you.